This is Science in Parallel, a podcast about people and projects in computational science. And I'm your host, Sarah Webb. For this episode, we're pausing our series on foundation models for an episode about using powerful computers to understand blood flow. I'm speaking with Amanda Randles about her work on building digital twins, personalized models of patient circulatory systems that incorporate data from smartwatches and other wearable devices. Amanda is an associate professor of biomedical engineering at Duke University. I've spoken with her several times over her research career. During Amanda's PhD at Harvard University, she started working on Harvey, computational model of blood flow that she and her research team at Duke continue to improve to this day. This interview is a longer audio version of a conversation that Amanda and I had earlier this spring and appears in the 2025 issue of Dexis magazine. Amanda was a Department of Energy Computational Science Graduate Fellow from 2010 to 2013. We often refer to that program as CSGF for short. It supports this podcast, and its fellows are meeting this week in Washington, D.C. as part of their annual program review. Amanda will be giving a keynote talk about her research. Join us to hear more about how she got her start, the latest on her blood flow research, and why she engages with the public and works on science policy. Amanda, it is great to have you back on the podcast.
Yeah, thanks for having me. I'm excited to be back.
One thing I've always found interesting about you and your scientific interests is that your training is in computer science and physics, but the questions you work on are very much about biology and medicine. How did you get interested in this intersection, particularly considering that your core background and training is really more in the physical sciences?
Yeah. So I guess I got started when I was in high school. I went to a math and science high school. So it was a math and science center where you spent half of your day every day outside of your high school with people doing programming. And that's where I first got exposed to computer science, learned how to program. We started with Pascal, and then we kind of moved into C. And that was kind of where, first got excited about what you can do with computer programming. But I really liked the scientific classes as well. I got excited about what you can do on the biological side. What are the biological questions? But I liked taking the physics approach and really understanding things from, first principles, fundamentals. So when I went to Duke for undergrad, I knew I wanted to study physics. I really liked the really understanding the nuts and bolts down to first principles of how does something work. I was planning on doing international relations. It was right around the time when the Human Genome Project had come out. So I was interested in how science affected policy at that stage. I was planning to double major in political science or international relations and physics. As a first year, some of my friends needed help with some of their coding classes. I spent a lot of time just helping tutor them and working with them, and realized how much I missed the computer science side. And I'm happy to stay up all night and pull an all-nighter coding. And I realized how much I missed that and how that was definitely something I enjoyed and wanted to do. So I kind of switched over. I had a double major in computer science and physics. I moved it to a minor in political science. And then in my first year, I was taking a biotech focus program. So some of our classes were in biotechnology, and then some were in the ethics and that side of it. And because it was right around the human genome time, they actually took us to the Craig Venter Institute, and they took us to go visit an NIH lab. And one of the big things that was coming out was how much data they were collecting from all the genome experiments. And they were kind of at that stage. This is back in 2005, 2006, where they were starting to sequence the genomes, but had so much data that, we're trying to figure out what do we do with all this data? How do we analyze it? And it started to become clear how much computer science could really affect answering biological questions. I got really excited about the role computer science could play in biomedical questions. I joined one of the genetics labs in my freshman year to start doing research, but all of the work, they did try to get me to do some of wet lab experiments. I was not very good at it, and it was not something I was very excited about. So every time they tried to get me to do those experiments, there was other researchers that would let me start playing with Perl and the bioinformatics research. And I kind of kept spending all of my time with the researchers doing the bioinformatics side. And because, wet lab wasn't really my thing, it allowed me to get involved with biomedical data, biomedical questions, but from like the computer science angle. I stayed in that lab for several years. I was always kind of involved with like the biomedical questions. I worked in a biomedical optics lab for a little while, but with that, I was always doing the computational analysis of the data that we were getting from those experiments. When I graduated, I was trying to decide if I should go into grad school or get a job. And then I was really lucky where I was offered a position at IBM on the Blue Gene supercomputer. And, they were using the Blue Gene supercomputer to study biomedical problems at a scale that you couldn't do otherwise. At the time, I was like, if I were to go to get a PhD, I would want this as my dream job afterwards. I'm being offered it now. I should just go take it and see what's happening. I spent a few years at IBM working on the Blue Gene team, and I had never really worked with parallel computing before that. So that was really the first time I saw what a supercomputer was, how you could use it. And then that kind of prompted me to want to go back to graduate school and focus on like how do we really use these supercomputers and how do we move it into something from the biological standpoint, which at the time I had talked to Tim Kaxiris, who is a professor at Harvard, and he was doing a lot of different studies in the biological space, but from the physics perspective and using models to understand biological problems and then using large-scale supercomputers. So it's a really good blend of all of my interests.
So let's fast forward to today. What are the main questions that your group is focusing on right now?
We're still using Harvey, which is like the code I've been working on since the Kaxiris lab at Harvard. I developed it there. I've kind of brought it through Lawrence Livermore and then back here at Duke. We're using Harvey to create three-dimensional blood flow simulations that are patient-specific. So a lot of the work lately has kind of changed into, not just personalized flow models, but really building a digital twin of specific patients, understanding their blood flow. We spent a long time, and probably the last time I talked to you, we were looking at really validating the model, so trying to replace invasive measurements. So one of the canonical examples has been for heart disease, you put a diagnostic guide wire in someone's artery and you measure the pressure across a stenosis or like a lesion where you might need a stent. And it's the pressure gradient across that stenosis that determines if you need a stent or if you don't. We developed this computational tool so you can make a 3D model of the patient's heart, run a blood flow simulation of their blood through that 3D model, and then instead of putting the guide wire in the patient, you measure the pressure gradient in that 3D virtual model. And then you don't have to have an invasive procedure. You just need the imaging and some data that we can collect non-invasively. We run a patient-specific flow simulation, and we conducted a large trial between Brigham and Women's Hospital in Boston and then Duke University. We compared the results from the simulation for 200 patients to the results with the guide wire and showed that you can accurately measure the flow and actually get this measurement, which is called fractional flow reserve, to determine non-invasively if the patient needs a stent or doesn't need a stent. So that's the basis of, we had an accurate model for understanding a patient's flow, like a patient's hemodynamics and their blood flow. And what we've been interested in lately is, we have kind of two major directions of connecting these blood flow simulations to wearable devices. So trying to not just understand your blood flow at one single time point for that diagnostic metric, but provide a remote way of monitoring how you're doing as you go about your daily life. And how do we move away from single time point metrics to, can we watch someone's blood flow over the next 6 months or over the next year and see if we can identify when someone's going to have heart failure or when they're going to have an issue before it happens, kind of like a check engine light for the heart, and help the doctor be able to act proactively and bring them in. So there's a lot of computational challenges there. It took the world's biggest supercomputer just to model a single heartbeat previously. And now we're trying to say we want to do 6 months of, many heartbeats. There's been like algorithmic challenges, computational challenges. We're kind of like, that's the area we've been focused on. At this state, we've been able to model 6 weeks worth of time, which is about 4.5 million heartbeats. The previous record had been about 30 heartbeats. So we've really developed a lot to expand that and make this possible to start looking at the remote modeling. And then on the other end, we're trying to understand, not just going in longer time periods, but higher resolution and scales. So we're trying to go down to, single cell model, understand why is a cancer cell interacting with the wall in the way that it is. So we've added adhesion models where we go all the way down to like individual ligand receptors of like, how is that cancer cell adhering to different parts of the body? The key problem is if you want to look at a large region of the body and all of the red blood cells, that takes a lot of computational power. Even using the world's biggest supercomputers, you can get about a 3 millimeter cube space and get all of the red blood cells in that space. And that is using the entirety of Summit, which was Oak Ridge's largest supercomputer a few years ago. And that got us a 3 millimeter cube.
That's tiny.
And we would like to look at much larger regions. So we had to develop new algorithms to allow, capturing cellular scale, but over centimeters or even a meter distance. So it was a lot of mathematical and algorithmic changes, physics-based changes, but then, allowing us to capture down to the ligand receptor pairings, but then capture the cell behavior and look at that adhesion over 10 centimeters and trying to get to that. So a lot of our work now is can we extend the time domain we're looking at and the scale and resolution that we're looking at, but over a larger space in that side of it.
Let's back out a little bit and talk about, digital twins themselves. I guess part of what I'm interested in is how close are we to applying the study that you just did where you compared the digital twin versus the current standard of care? How far are we from basically switching people over to this less invasive type of procedure and having that be routine?
For the study that I mentioned, not our tool at this stage, but there are similar tools out there that are already FDA approved and being used in the clinic regularly. So for single time point metrics for certain diseases, particularly for a fractional flow reserve, there are several FDA approved devices being used for non-invasive measurement using digital twins or personalized models for these kinds of flow simulations to decide if you need to have a stent or not. They vary from one-dimensional models to 0D models, and they also have some 3D heavy flow simulations. And they're already being used in the clinic. There are even a few tools that are being used to predict for treatment planning. And we've been working on this as well, where it's using machine learning to say, if I were to put, stent X or stent Y in, how would that affect the flow for that patient? Or if there are multiple lesions in the patient, which one should I be stenting? So there are a few approved tools already being used for some of the treatment planning. That's mainly in the heart. We need to see that in other areas of the body and the vasculature. So it'd be great to be able to use that in the leg or in the brain and other spaces. It's less mature in some of those spaces, or at least the translation of it into the clinic hasn't been seen as much. But a lot of what you see in the clinic today is really tied to a single heartbeat, a diagnostic metric, which also honestly, sometimes we call a digital twin. It's kind of on the border of is it really just a personalized computational model? I think if these models start to become digital twins when they're connected and you're getting real-time feedback from the patient and you're tying the physical model to the virtual representation, and it's not a disjoint process. And that's when it becomes a digital twin versus just a personalized model. And that's where tying it to the wearables is really moving us to this idea of the digital twins. And I think we're really starting to see that happen, but that's still in the research phase. And I think we're still a few years away and there's more studies that need to happen, because for studies like the vascular digital twins, we've only just been able to model longer flow periods. And now we need to do all the clinical studies to show that it's accurate over time. We've never been able to access these fluid dynamic metrics continuously over long periods for these patients. So I think there's even new markers that we don't know are going to predict heart failure, but we need to do all the studies, the discovery, to find out what are these markers and then show that they can be valid in the clinic. So all those kinds of studies are in the process.
These are huge simulations. And you were talking about how important supercomputers have been to how you think about problems and the problems that you've worked on. And you've been a part of the early work with Aurora. So what makes supercomputers such as Aurora essential for your work? And what have you been able to do on that system so far?
The discovery phase is really important right now. So we don't necessarily know what the biomarkers are. We don't know what we're looking for. And so with that, we need to collect everything and then figure it out. The goal would be, you can create a reduced order model, or in today's world, we can now create these AI surrogates so that when you translate it to the clinic, you don't need a big supercomputer for every single person. But until we know what we're looking for, we need to run incredibly high resolution simulations. We need this kind of capability. For us, the first time we ran a coronary study I think it was like a 10 micron resolution. So at the resolution of a red blood cell, it took the entirety of the Argonne's biggest supercomputer back in 2010, which is like 140,000 processors. But it took us 6 hours on the whole system just to run one heartbeat. It is important to not just capture one vessel of the coronary arteries. We know that having the side branches and the entire geometry is really critical to understanding the flow patterns and what's going on. So we can't just limit the geometry, but we also need a really high resolution, and we want to be able to capture it over time because the pulsatility of the flow and how it changes is incredibly important. But how often are you going to get the entire supercomputer? So even just waiting for the supercomputers to get bigger really isn't enough. In 2015, with the Livermore system, we were able to go to the level of like the full body, but it required the entire supercomputer. And it wasn't even the full body. It's like the scale of the full body, but it's all vessels that were 1 millimeter in diameter and above. So you're not even capturing anything below that. And that still required the bulk of the supercomputer. Now with Aurora, we can run that full body simulation only on like a smaller portion of Aurora, which really allows us to run many of those simulations and do more exploratory science of like if you're going to change heart rate or change the heart rate variability or other characteristics that we know are associated with disease development. Can we do these parameter studies and see how they affect the pressure in the patients and really try to understand what's going on with that patient? If we want to move to the digital twin side where we're trying to capture 6 months of time, we need tons of compute hours. And then on the cellular components, we're now with Aurora, we've been able to model at least a few billion red blood cells. It's only a small fraction of the red blood cells in your body, but it's a huge amount to really understand the interaction of billions of cells at a time and really, how a cancer cell might be moving through the body, where it's interacting with the red blood cells, where it's moving is really critical to capture these cell-cell interactions. And Aurora is allowing us to fully deformable red blood cells that are adhesive and really mimic the physiology of the system without having to make as many assumptions. So it's been really helpful, but it's very computationally intensive. And then it's also resulting in tons and tons of data, like terabytes to petabytes of data being created for all of these simulations. What do we do with this data? We've been working with the Argonne team very closely of just how do you visualize that amount of data? So we're trying to do a lot of like in situ visualizations. So trying to visualize the cells while everything is still in memory on Aurora and like ways to access that because you can't really download a petabyte of data, take it to another type of system and try to visualize it there. So there's a lot of questions aside from just how do you make the simulation, but then how do you just analyze the data that comes out of that simulation?
Within your team, I mean, it sounds like there are so many biological questions that you're thinking about, the fundamental fluid dynamics of what's going on.
Yeah.
The algorithmic questions, the visualization questions. Within your team, do you have people who kind of gravitate towards certain problems with something that's so interdisciplinary? How does that come together?
It's kind of amazing of like, the people on my team are really like, they're very ambitious. They're willing to take on hard problems. They're all incredibly smart. They all have to know a little bit of all of the pieces. You can't just have like your fluid person who then talks to the computer science person. I always feel bad because like when they join the lab, they have to learn the lattice-Boltzmann method that we use. They have to learn a little bit of high performance computing. Most people are all in biomedical engineering, so they're doing all of this to answer a biomedical question. So they need to also get to that stage. So they have all of this base knowledge. And then different people in the lab, as you're saying, like kind of gravitate to different pieces. So we have one person who is, while his thesis is really focused on adhesion and like cellular interaction, he's also the person who has ported our code to every imaginable programming model and is, amazing on the parallel computing side and is testing the efficiency. And then along the way is getting like supercomputing papers on the portability and efficiency of the code while trying to do all of this so that he can answer a question about cell adhesion down the line. So the students are very interdisciplinary and they kind of bring all those skill sets and they kind of focus on making these computational advances so that they can answer their biomedical question. We have people that end up focusing on different areas. The lab is kind of separated into key project areas and we kind of meet in like mini teams to get these pieces together. But then in the group meeting, we have a part of the meeting where you're kind of reporting back from every team. And there's part of it that's like, what are the points of discussion that other people need to know of? So if you developed a new technique or something new, you can measure a way of speeding up the code that might be useful. If you're on the cell team, but it would be helpful for the heart failure team, and kind of report that back in the group meeting and share between. So you'll notice like my publications, there's never just me and one person. It's a very team-based science and works really well.
So I'm going to shift gears a little bit because you're definitely out there talking about your work. You're active on social media. You've done interviews with science journalists, and you're participating in policy discussions about the future of computing. Why are these conversations beyond the lab important?
It's really critical for people to understand, the kind of work that we're trying to do and like the role that it can play and why kind of on multiple fronts of there's a lot of work with AI and digital twins and all that in the biomedical space. And it's important for people to understand like what's really happening, what's coming from the experts of like, how are we using it, understanding the impact on security, the impact of like, how is this really going to help you? What is the benefit of this kind of research? And then what can you tangibly expect to see? Like you were asking earlier, like what's going to happen in the next few years? What is the difference between science fiction, what we think is going to happen, and like what is really going on? And kind of helping people understand what's available now, what can you make use of now, even just in terms of, the data we get from wearables, the data that you can access on your own. And like, I think as we're increasing from the biomedical side, we're kind of at a shifting point with wearables and with the way patients now have more access to their data and they can, see what's happening with their physiological state. We're kind of moving from the point where all of your information is owned by the doctor and in your medical records. It's more empowering for the patients. It's important for the patient to really understand what are those opportunities and how do you actually make use of this data and understand what's out there. It's critical on that side. On the HPC front, there's obviously a lot going on with, AI development. We've kind of come to a world where like everybody even actually knows what a GPU is, which is pretty cool. But not everybody understands what are the nuances of what does that mean. One of the issues we have is there's a lot of development for AI-based GPUs, which is fantastic, but potentially a lot of like the architecture that's being created to really support some of these AI algorithms may not be what we need to support physics-based modeling, large-scale high-performance computing. And we kind of need to figure out like what is the path to kind of maintain those types of specialized hardware, but also having it come in a way that is going to be useful for these large-scale models and why these large-scale models are important, what we can get out of them, and making it clear. We used to have through the DOE, the big exascale project, which really helped us move forward technology for the space, not just the hardware, but the software and training for people to be able to use these systems. We don't have a plan going forward after the exascale project. So a lot of it is, what do we do from here and how do we maintain that and improve on it? How do we build a workforce that can use this technology and use it intelligently? I think it's an important conversation to be having with policymakers, with the public, and kind of informing on all ends.
You were a co-author with a number of other computational scientists in this recent policy article in Science about the need for these long-term plans and technology leadership in HPC. What right now would you say are the critical needs in this area?
My involvement with this article came out of one of the committees I was helping lead through the DOE, where we were trying to look at, what is the future for the large systems? What's the path for Oscar? What is their 10 to 15 year plan? And where would they be going?
Oscar is the acronym ASCR and stands for the Advanced Scientific Computing Research Program within the U.S. Department of Energy's Office of Science.
And every conversation we had, it kind of just kept coming back to, it's great that, we're trying to get this plan in place, but we need so much more than just the hardware. There's so much more that needs to happen. And we kept having all of these conversations and it kind of snowballed into, well, why don't we just write this other article? The key piece is there's just so much that needs to happen. It's not going to be able to be funded by just the Department of Energy creating a large supercomputer and putting it out there. We need a whole government approach. It's this interdisciplinary problem that needs to be solved and needs to be attacked from all angles. Like we need the workforce that is going to be trained to be able to use these systems, but we need systems that are going to be able to address pressing problems for the nation and understand what those problems are. It's not all just AI, and it's what kind of models do we need to build, and then what kind of systems can actually support answering those questions. It's not just health and digital twins, but it's like we have different architectural needs than you might have for other types of workloads. And we need to assess, we need to be able to have a plan for how are we going to ensure strong development of new technology, not just the chip, but also the interconnects the entire system. But it's also really important to have support for the software stacks and not just like the application level, but like all the way through. And we really need to have the exascale program really supported the software stacks and development of like the libraries that we need and the underlying like the mathematical libraries and like the support on that end. And we need sustained support for those kinds of libraries and that kind of software support to make those usable and kind of modify to all the new architectures that come out. And currently there really isn't a clear plan in place for how are we going to have long-term sustained support for development on all of those fronts. And I think that it's critical that we have these conversations, that we plan early, and that we start trying to figure this out.
You've been talking about workforce. How has the DOE CSGF influenced your career?
CSGF, I think, is amazing, and it had a huge impact. I'm still heavily involved with the CSGF itself, but I'd also say a lot of my collaborators, a lot of my mentors, I've gotten so much out of CSGF aside from just like the original funding that gave you that flexibility to do the research you really wanted to do. I'm not sure I would have been able to develop Harvey in grad school and have that flexibility because it was very much a side project for my advisor. I think having CSGF in the first place gave me flexibility to pursue the research I was excited about. And then I was able to do my practicum at Livermore, which I then went back and did my postdoc at Livermore. And it really set up those interactions. And I got to meet, it introduced me to the mentors and allowed me to pursue research and set up research programs like I don't think I would have otherwise been able to do. When I was a grad student, I had many mentors that helped me figure out the next stage of my career that really came from the CSGF program. Even still, I go to people like David Keyes, who's heavily involved with CSGF, and ask him for career advice. He still writes letters for me because of our first interactions at CSGF. It really helps you have a network of people outside of your graduate program and introduces you from your cohort to the alumni and like the advisors involved. It just creates a real community. As opposed to other fellowships where you just get funding, they get you together every summer. You really bond, you get to know each other. It's been a really fantastic experience for me. Right now, we have a virtual seminar series that we're setting up at Duke. We have a new center at Duke focused on computational and digital health. We've had a lot of like former CSGF speakers coming in, and there's a lot of connection with CSGF through that.
What pieces of advice would you give to early career researchers, say current CSGF fellows, other up-and-coming computational scientists?
I think there's a lot, especially like having just mentioned David Keyes, it's like be comfortable, especially like within CSGF and other places, reach out and ask for help and like ask people for advice. I know like we're all busy and we may not be able to respond, and don't be offended if people don't respond, but it's worth asking. Like, I know when I was in grad school, I was trying to figure out what to do after my PhD, and I was very stressed out. David happened to be giving a talk at Harvard that day, and I just like emailed him and was like, I'm really stressed. I need some help. What do I do? And he met with me after his talk. And in the meetings with David, he actually like helped introduce me to people who helped, I had job interviews based on that discussion. He's been, a resource of bouncing ideas off of him and trying to figure out career steps. And it's just feeling comfortable to reach out to people and ask for help because a lot of, we've been through a lot of these things. We all understand that it's stressful. I think the job market is even getting more stressful. Ask for help. Every time I've been changing jobs and doing things, I've used alumni networks through CSGF or through the universities I've been at to try to find jobs, reach out for informational interviews, and just it's okay to ask for help if people have been through it. And then on the research front, I would just say don't kind of bend to what should be funded. Think about like what really motivates you, what you're most passionate about, and kind of be willing to take big risks and try crazy projects. Because I think a lot of the more fun projects are the ones that are really like at these interdisciplinary boundaries and are high risk, high reward projects and kind of might seem crazy at a first pass, but a lot of interesting things can come out of it. Keep trying and don't be afraid of failure. And, failure is definitely a key part of this. And we all have it all the time and just be willing to take a risk and try different projects.
Great. Amanda, it was a pleasure as always. Thank you so much.
Thank you.
To follow Amanda Randles and her lab on social media and to learn more about the people and projects mentioned in this episode, please check out our show notes at scienceinparallel.org. Amanda was also a guest on the podcast in 2022. I spoke with her and another former CSGF recipient, Anda Trifon, about the interface of computing with biomedical research, including their work on COVID-19. Science in Parallel is produced by the Krell Institute and is a media project of the Department of Energy Computational Science Graduate Fellowship Program. Any opinions expressed are those of the speaker and not those of their employers, the Krell Institute, or the U.S. Department of Energy. Our music is by Steve O'Reilly. This episode was written and produced by Sarah Webb and edited by Susan Ballett.